everyone. My name is Hiroshi Ezura. I'm a CTO of the Sanatech Seed Company and also a professor of the University of Tsukuba, Japan. And today I'm going to introduce our innovative crops, which is a high-GABA gene-edited tomato, which is the first genome-edited tomato to be commercialized all over the world. Sanatech Seed is a venture company spinned out from the University of Tsukuba. In 2018, our company was founded with the purpose of protecting the children and us of tomorrow by using biotechnology innovations to improve agriculture crops. The term Sana used in our company name is a Latin word which means health, and it presents our wish to improve people's health through our innovative products. Okay, so this is our first innovative tomato developed by using genome editing technology. The name is Sicilian Rouge High-GABA. It's the first, which is the first genome-edited crop commercialized in Japan, and also the first CRISPR crop commercialized all over the world. And we are started commercialization of these products in May 12 this year in Japan, and this is our first commercialization all over the world. So in this slide, I would like to explain what is GABA. GABA is a gamma aminobutyric acid, which is a non-proteogenic amino acid, and found in animals, plants, and bacteria, and involved in the biotic and the biotic stress responses. But for human, GABA function as a neurotransmitter, and because of this function, when we orally takes this GABA, which can reduce the blood pressures and also induce a relaxation. Because of these functions, this GABA has been used as a food additive in Japan and produced a different kind of health-promoting food, as you can see here in the slide. And then now GABA is a very famous health-promoting functional compound in Japan, which is like a vitamin C. So the Japanese people know this what is GABA well. Okay, now I would like to introduce partly the gene editing technologies. This technology is to induce a mutation into the target gene, as you can see here. And so far, there is several kind of genome editing technologies developed, such as the CRISPR-Cas9 and the TALEN. And these tools introduce mutation into the target genes. And during the repairment, in the most case, this break has been accurately repaired as a normal sequences, but sometimes the mutation happen. The mutation are induced during the repairment, and which create deletion mutation and the replacement mutations and the insertion mutation. And those mutations alter the phenotype of our target crop. So this is gene editing technologies. And then here you can see the most famous gene editing technology, which is the CRISPR-Cas9 technologies. This CRISPR-Cas9 technology is CRISPR-Cas9 composed of two components. One is a Cas9 protein, and another component is a guide RNA. That these complex make the DNA break at the target site. And then this guide RNA bind to a target site, and then Cas9 recognize the NGG sequence, which we call a PAM sequence, and then Cas9 cuts the 3 bases upstream of this PAM sequence, and then as a third step, the repairment of DNA error occurred in the first third step, and those error becomes a mutation in the genes, target genes. Okay, this Cas9, CRISPR-Cas9 was developed in 2012, and then 2020, it is the researchers developed this Cas9, awarded a Nobel Prize in Chemistry. And one is Emmanuel Charpentier, and another one Jennifer Doudna, and they obtained the Nobel Prize in Chemistry. And they say that this CRISPR-Cas9 technique will be expecting that it is very useful for crop improvement. So here and then, so that's why there are many scientists who are involved in the plant breeding try to start using this CRISPR-Cas9 technique for the crop improvement. And here you can see the summary of the merit of gene editing technology for the plant breeding. One is which is this technology is efficient, become efficient breeding techniques. That means that we can reduce the effort, times, and cost for breeding. And the second merit is effective utilization of genetic resources. Because of this merit, so the scientists intensively start using this new innovative technology in the plant breeding. Okay, in this slide, I would like to introduce what is the difference of GM crop and the gene-edited crop. Sometimes two words is very confusing, but there is a clear differences. Okay, so for a GM crop, where we introduce additional gene to create a new trait, but genome editing, we induce a mutation into the endogenous genes to modify the phenotype. So this is a very different point between a GM crop and the gene-edited crop. And also, compared to the conventional breeding, the GM technology create a new trait which we cannot confer by conventional breeding techniques. But by GE crop, comparable to the mutants created by traditional mutant breeding. So that's why the products of GE, some product of GE considered to be same with products by conventional breeding techniques. And then another more clearly, you can see the difference. The target genes is the final products in GMO, GM crop. It contains additional genes which can introduce other organisms. But in G crop, there is no additional gene, just a mutation created in the endogenous gene. This is a very big differences. And based on these differences, the evaluation for food safety assessment and also environmental risk assessment has been is different with the GM crop and the G crop here. More precisely, it should be more clear by this slide for clearly show the difference of GM crop and G crop. So here you can see the tomato. The tomato has 34,000 endogenous genes. But for gene editing improvement, we initially introduced gene editing tools, genes. But in the final products, we delete the gene editing tool. So the gene number of the before and after the breeding is the same. The number is 34,000. But in GMO, and we add the more than one genes. Well, in case of tomato, if we add the one genes to provide disease resistance, so number of the genes in the final products increase to 34,001. So that's why we need to carefully evaluate the increased the gene for environmental and food safety assessment. Okay, so in this slide, I would like to introduce how unique our product is. So by using genome editing technique, we increase the GABA content in the tomato fruits. And here you can see the difference of GABA content in tomato. As I mentioned in the previous explanation, GABA included in all crops, but the amount is different depending on the crop species. In this slide, here you can see the white bar show the original tomato line, the GABA content of original tomato line, and the dark bars show that the GABA content of genome-edited tomato. As you can see here, we measure the GABA content at the mature green stage and the breaker stage and the red ripe stages of fruits development where we harvest the tomato fruits. As you can see here, GABA content of the gene-edited tomato is 4 to 5 times higher than original cultivars. And this 4 to 5 times increase of GABA is enough to expect the function of GABA, which is like lowering the blood pressure and also make a relaxation. So this is the most important difference, significant difference with our gene-edited tomato compared to the original tomato cultivars. So then we can eat just a small amount of tomato fruits, like 20 grams or 30 grams, which is 1 or 2 small cherry tomato fruits, as enough to expecting the lowering to get blood pressure and also make a relaxation. Okay, so from here, I would like to introduce the regulation of gene-edited crop in Japan, and then we'd like to introduce how we can overcome the regulation in Japan to sell our products. So here you can see the category of gene editing technologies. Now, gene editing technology has been divided into 3 types, 3 classes. One is SDN1 and SDN2 and SDN3. And the SDN1 is just cutting the target gene, and the repairment is naturally repaired. this is almost similar with conventional mutagenesis, like a chemical or physical mutagenesis products. So that's why in Japan, these products developed by SDN1 technology considered to be a non-GM. So then we can grow anywhere and we can eat as a food. And but if we develop the gene-edited crop using STN2 and STN3 technologies, some of those are considered to be GMO, and sometimes GMO or non-GMO, depending on the ministry. Now we are still discussing about the position of products of STN2. But today, in our development, I only use STN1 technology, so that our products should be considered as non-GM crops in Japan. Okay, so here you can see the steps, commercialization or deregulation of gene-edited crop in Japan. So developer, in this case, for instance, a Sanatech Seed, developer have to get a prior consultation with two ministry, Ministry of Agriculture and Food and Fishery, and the Ministry of Health and Welfare. So when the developer develops their genome-edited products, they provide some information to evaluate the environmental safety and also food safety to the two ministries. And then after the completion of this prior consultation, then developer can complete the notification to the two ministry. And then after the notification, we can use products other like normal crops. Okay. During the here you can see more detail about the prior consultation with Ministry of Health and Welfare for food safety assessment, and so developer have to get a prior consultation with the Ministry of Health and Welfare, and then this ministry have the committee on to evaluate the food safety issues on the products, and then they evaluated the data we presented, and then if they have some question, then the question come back to our developer, and then we repeat this prior consultation cycle, and then we can answer all the questions to the committee, and then so we can complete the notification to the ministry. And then we can start the commercialization, or we can start eating of this gene editing crop. So this is we did, and during this prior consultation, we have to provide this kind the information listed here 1 to 6, and the most important point is that we need to show the data, the absence of transgene, because when we are conduct gene editing technology, we once introduce a gene into the genome, but in the final products, we deleted the tools from the final products. That we need to prove the absence of these tools in our products. So that is the most important information. And also we have to provide the information of allergenicity, and also the change of the toxin, natural toxin content. As you know that the tomato contain a natural toxin like a tomatin. So then we have to we measure the tomatin content in our products, and we provided those data to the ministry, and the committee member evaluated the content is different with the original one and our gene edited one. So we provided such kind of information to the committee. And that is the same into the environmental safety assessment. We have to provide such information to MAF, Ministry and Agriculture Fisheries, and those information we have to provide it to the ministry is listed here, and almost the same. But and also we need to discuss the on possible influence in biodiversity, biological diversity when the organism is used or that means that when we cultivated our gene-edited tomato in the field, make some discussion about. But anyway, so after submitting and this information to the ministry, and then as you can see here, we got a certificate for which show they are confirmed the environmental safety and food safety and feed safety issues for our products. And then we got this certificate, and then we can start the commercialization now, and which is we finished the completed our notification December 11 and 2020. Because this is the first products, and we spent almost 1 year to complete a prior consultation. Yeah, so then we could now we are at the stage of commercialization of this new innovative products of tomato. Okay, so after the completion of the notification to Ministry of Japan, then we could confirm our product is safe and environmentally safe, and also safe as a food. But when we start commercialization, we discussed about the how to provide the information to the consumers. That is the most important information. According to the Japanese regulation, we don't need labeling is not a mandate in Japanese regulation, because they decided that gene-edited crop they evaluated is almost the same with products by the conventional breeding techniques. So then they don't need to ask to labeling. But our company decided to make a labeling because of this is the first genome-edited crops commercialized. So then by labeling, we decided to providing this information. This tomato has been improved by genome-editing technologies, and also the notification to ministry has been completed. So our consumer can get the information of what they are eating. So this is a very, very important point, and for the fast commercialization, so that then consumer can confident about our products. Okay, here you can see the traditional value chain of agricultural products, and seed companies. They develop a seed and they provide the seed to a farmer, and the farmer grow the products and harvest, and the supermarket collected those products, and the supermarket sell the products to the consumer. So this is the traditional value chain. But we thought that if there is even a small number of consumer or people is concerning about this new technology and new products, so maybe farmer don't want to grow, and also supermarket wanted to handling or sell these products. But I already involved in the development of genome-editing tomato crops for almost more than 5 years. And then I made a workshop and a webinar for the consumers and the different stakeholders. And then I feel that there is many people or many consumers actually wanted to have these new products. So our challenge is how to deliver our products to a company, to a consumer. Okay, so we decided to bypass the gatekeepers like farmers and supermarket as an initial commercialization step. So we try to directly deliver products to the consumer, and bypass the gatekeepers. And also we try to establish a bi-directional connection or communication with the consumers, because we wanted to hear the direct voice from the consumers. And then we try to make a easier consumer to access our products, which is okay. So our consumer, and then among the consumers, we are initially targeted home gardeners. In Japan, the home gardening is a very, very popular activity for the consumer among the consumers. And so we decided to, as an initial step, we targeted home gardeners, because a home gardener has a two function. They produce products by themselves in the garden or some small field, and also they can eat their products by themselves. So they are consumers. So we named them as a prosumer. So they are two. So if we can provide our gene-edited tomato to these home gardeners, so then they can grow and they can feel and they can understand what is a gene-edited tomato. So we decided to provide deliver the products to these kind of home gardeners. And then also as an initial trial, we decided to provide free samples to the home gardeners throughout Japan. And so by then we could receive their unfiltered and authentic feedback directly to via the following social channels. And it was very, very fun for us and useful for both sides. And after the completion of the notification to the government, December 11 in 2020, we announced that our company is going to provide a free sample growing kits to the home gardener. And surprisingly, amazingly, we had many applications for growing kit. So we had more than 5,000 applications for this growing kit. And eventually we provided 4,200 samples kit to across Japan, all area in Japan. And then we also provided a consultation of how to grow this tomato, and also provided a growing guidebook to the consumer. And during this activity, we can communicate with those consumers directly, and we can get direct invoices from consumers. And then now we are trying to integrate those consumers' opinion to our next development. And but here, this is a very, very amazing for us. We are not expecting such as whether we can get such a huge number of application to our tomato. And here you can see some example of home gardeners growing our GE tomato in their garden. And we provided the free sample seedlings in this May 12 start. And then some grower growing the tomato, as you can see here, and then they harvest the fruits, and then they enjoy these red fruits. And we have a lot of many positive feedback from the home gardener who grow our High-GABA tomato. During the communication of home gardeners, we have requested about to sell our fresh tomato during the winter. As you know, the winter in Japan is very cold. Then the home gardener can grow the tomato in their garden. But after experience our High-GABA tomato, they wanted to continue to eat during the winter. So that's why our home gardener requested us to provide the fresh fruit even in the winter. So we decided to find out a contracted farmer and ask grow our High-GABA tomato, gene-edited tomato, in their greenhouse even in the winter. So we started the cultivation of a High-GABA tomato at a contracted farmer from this summer. And then we started selling our fresh tomato to the consumer from on this September 15th. So this year, the September 15th is a kind of a memorial days, which is actual day for commencement of actual commercialization of gene-edited tomato. And then our company continued to selling the fresh fruits to even in the winter. And in addition, we have another request from our consumers or home gardeners. They almost now we are in Japan is winter, so they almost finished the cultivation this year. But they wanted to grow our High-GABA tomato even the next year. So they asked to provide seedlings for the next year, and then we start booking for the growing kit for the next year in this October, and also our home gardener, they also asked to prepare some processed food using High-GABA tomato. So then our company now developed the some processed food, and then going to selling those processed food with this our High-GABA tomato in next maybe January. So this is our next step of commercialization of gene-edited tomato. And then we have a many order of fresh tomato and also a growing kit and even the processed tomato so far. So I think that at the initial stage of the commercialization, the situation is pretty good. And we are expecting the next year those kind of requests or selling will be more increased in the coming year. So in this marketing, we use a D2C marketing system, and where we only grow. We grow only contracted farmers, and then all purchases all produce, and then we produce only not only a fresh tomato and also processed products, and then we are going to sell it through our website, and then we are as an initial stage we are targeting the consumers about high blood pressures and also lack of sleep. So the consumers who are looking for a functional food, so-called functional food. Okay, so advantage of this D2C marketing, there is many advantages. First advantage is consumer can directly buy anywhere, anytime. They can buy our High-GABA tomato anytime, anywhere. And then for the companies, we can directly communicate with the consumers. So then we can hear the actual voice from the consumers. And also we are asking only a contracted farmer to grow our products. So that can make we can show the traceability of our products. That means that we can handle all products by our companies. So this is many advantages of D2C marketing system. So maybe this kind of functional food will be contributed to not only a developed country and also but also in developing countries. So this is our motivation why we try to increase the functionality of the food. So actually in my laboratory we spend to apply genome editing techniques we need a basic research. So we spend almost 10 years to understand how tomato accumulate GABA in the tomato fruits. And here you can see that some of the results. Here you can see the GABA metabolism in higher plants and in tomato. And then we find the key genes for regulating GABA accumulation in tomato fruits through our basic research. And then we identify that this gene, GAD genes, GABA, which GAD3 gene, which is key genes regulated GABA accumulation in tomato fruits. And then we did some biochemical analysis for this enzyme, and then we find out that by introducing a small mutation into this gene, that can increase the activity of this enzyme, and then that's resulting in the GABA accumulation in the tomato fruits. And then we did a lot this kind of basic research, and then in the 2012, the new technology, CRISPR-Cas9 technology has appeared, and then we decided to use this technology for the improvement of GABA accumulation in tomato fruits. And we introduced a point mutation to GAD3 genes using a CRISPR-Cas9. And then here, this is a proof of concept experiment. Wild type is the original content of original GABA content in original tomato. And then other lines show the GABA content engineered by gene editing CRISPR-Cas9 technology. As you can see here, the GABA content in the tomato fruits has been completely increased to original tomato cultivars. So here you can see the history of GABA, high-GABA tomato history. We started the development basic research in the 2005, and they spent almost 10 years to see which is the target gene, and then how we can introduce a mutation, and that's resulting in the increase of GABA content. We spent almost 10 years. Then we actually did a proof of concept experiment using a model tomato cultivar, and then we established the genome-editing technology to increase the GABA content in tomato fruits in the 2017. And then 2017, we decided to use this new technology to improve the actual commercial cultivar. So then we established a venture company, Sanatech Seed 2018. And then this Sanatech Seed start developing a commercial cultivar. And then finally, this year, 2021, we could start the commercialization of products. Actually, engineering itself, we spend just 3 years. It's very, very efficient compared to the conventional breeding techniques. So this is the history of high-GABA tomato. Okay, so for me, the productivity is a quick decision, and also the maybe challenge is productivity. So when I start actually this development of gene editing crop is started as a national project of Japan, supported by Cabinet Office of Prime Minister in Japan, and many researchers are involved in these products, but most of them wanted to commercialize their products for the consumers. But in our case, by establishing venture companies from the university, and but it's a small company, but it's very, very quick, and we can make a very quick decision, and then so that is a key point of the productivity of the research in the future. Okay, thank you very much for your attention. And so this slide is my final message. Now, a new era of rapid and precise breeding via genome editing. So genome editing allows an order-made breeding for a sustainable nutritional food production. Then we should use these new techniques for our future. Thank you very much for your attention.